The branches of math and science are often taught separately. Most students don't use differential calculus to calculate the rate of DNA replication, or use chemical kinetics to represent repressor binding to Lac Operons. But this past year, there was a question on my Harvard problem set that was so beautiful, so intricate, that I just couldn't help but share it with all of you. Now, a quick disclaimer. The question that I'm about to show you is actually not the exact question that was on my homework, and this is because there are strict rules against posting homework questions and answers online. However, I created a variation of the problem that requires the same set of skills to solve. So, the following question was written by me, but if you're able to solve this question, then you essentially would have been able to solve the actual Harvard homework problem. So, ready? Here we go. You are studying eukaryotic transcription factors alpha, beta, and gamma, whose production is part of a biochemical pathway as depicted below. Every step in this pathway is a first-order reaction, and there are no intermediate steps in between. Alpha is converted to beta, which in turn is converted into gamma. Gamma, however, is known to be unstable and thus degrades back into alpha and beta. In order for a given cell to express gene X, it needs 8 molar of transcription factor beta. Given that your cell of interest begins with 14.98 molar alpha, 0.01 molar beta, and 0.01 molar gamma, will gene X eventually be expressed? If so, how long will it take? And if not, what's the maximum concentration of transcription factor beta that's produced in your cell of interest? Now, pause the video and take a few minutes, see if you can solve this on your own. Okay, let's break this question down. We're first introduced to 3 transcription factors, alpha, beta, and gamma. If you're familiar with biology, then you'll know that transcription factors are small proteins that help turn genes on. Basically, in your body's DNA, there are many important segments called genes, such as those that determine how tall you'll be or when to make keratin, which is in your hairs and nails. These genes can be turned on or off, and a major determinant of whether or not a gene is on is the presence of transcription factors. Our question later explicitly states that gene X requires 8 molar of transcription factor beta in order to be turned on or expressed. So our goal is to determine whether or not gene X will be turned on based on the concentration levels of transcription factor beta. Essentially, will at any point in time the concentration of beta be equal to or greater than 8 molar? And if so, when? However, even though our end goal is to determine whether or not we will have enough transcription factor beta, we must also look at alpha and gamma, because they're all related to each other through a chemical reaction system, where alpha forms beta, beta forms gamma, and gamma decomposes back to alpha and beta. If you're familiar with reaction kinetics from chemistry, then you'll know that we can write rate laws that tell you how fast a reaction is proceeding, as long as you're given the chemical concentrations and the rate constants denoted by lowercase k. For instance, for the following reaction, we can write the following rate law: K times the square of the concentration of nitric oxide times the concentration of oxygen. And notice that these brackets mean concentration of in chemistry. As you can see, the coefficients in the reaction are the exponents in the rate law. An important thing to keep in mind is that you are only allowed to do this when the given reaction is the rate determining step of the overall reaction. For example, the chemical reaction nitrogen dioxide and carbon monoxide forming nitric oxide and carbon dioxide actually consists of 2 intermediate steps as shown below. So the rate law is not equal to K times the concentration of nitrogen dioxide times the concentration of carbon monoxide, but rather K times the concentration of nitrogen dioxide squared. In our question, we are explicitly told that there are no intermediate steps, so by definition, every reaction in this pathway is a rate determining step, so we are allowed to write rate laws from the concentrations and rate constants. So now, let's try writing the rate laws for the formation of transcription factors alpha, beta, and gamma. If you're familiar with differential equations, then you know that we can write the rate at which the concentration of alpha changes over time as d alpha dt. Thus, we can write d alpha dt equals the rate of alpha formation minus the rate of alpha loss. And by combining our knowledge of chemistry and differential equations, we can ultimately find that d alpha dt equals 3 gamma minus 2 alpha. And if we repeat this process for transcription factors beta and gamma, we get this system of differential equations, which we can rearrange like this. But now what? This system of differential equations only tells us the rate at which the concentration of transcription factors is changing, but not the actual concentrations of the transcription factors themselves. How do we know if the concentration of beta will ever be 8 molar if we're only given the rates and not the actual concentrations? Well, here's an idea. Intuitively, we know that this chemical reaction system will eventually reach dynamic chemical equilibrium after a long period of time. Chemical equilibrium is when the concentrations of all species, in this case alpha, beta, and gamma, no longer change over time. They stay constant. So if a chemical reaction system is at equilibrium, then even if we wait, let's say, an hour, there won't be any more or less transcription factor beta than before. Therefore, by definition, equilibrium is where d alpha dt, d beta dt, and d gamma dt all equal 0, a property that we can use to our advantage. A quick aside, in biology, this phenomenon is actually more accurately referred to as steady state rather than equilibrium, but that isn't too important for this video, so I won't get into it. So, how will we use the idea of equilibrium to our advantage? Well, let's say that we perform our calculations and find that at equilibrium, the concentration of beta is 14 molar. Well, since we started with only 0.01 molar of beta at the beginning and managed to get 14 molar of beta sometime later at equilibrium, that means that the concentration of beta must have been at 8 molar at some point in between. We will use this principle, which in calculus is called the Intermediate Value Theorem, to see if there will be 8 molar of transcription factor beta at any given point in time. So let's solve for the equilibrium concentrations by setting d alpha dt, d beta dt, and d gamma dt equal to 0. And by doing that, we get this system of differential equations. Now, you're probably thinking, oh, we have a system of linear equations, so we can just easily solve for alpha, beta, and gamma now. And that's a great idea, we can try that. But if you try that, you realize that you can't. See, if you do the math, the closest you'll actually get to the values of alpha, beta, and gamma are the relationships alpha equals to 3/2 gamma and beta equals 5 gamma. But you won't be able to find the absolute values of alpha, beta, or gamma, just given the information that we have so far. We need one more piece of information to solve this. And we actually have it right in front of us. In the original question, we were told that we started out with 14.98 molar alpha, 0.01 molar beta, and 0.01 molar gamma. Now, at first this might not seem very useful since we're studying the reaction system at equilibrium and not at its initial value. But if you think about it, based on the law of conservation of matter, the total concentration of species in this reaction shouldn't change over time. Thus, in total, if we add up these initial concentrations, we know that we start with 15 molar of these 3 transcription factors combined. And by the law of conservation of matter, we know that we'll always have 15 molar of these transcription factors. So even at equilibrium, we can say that alpha plus beta plus gamma equals 15. And thus, by using this information, we can then calculate that at equilibrium, alpha equals 3 molar, beta equals 10 molar, and gamma equals 2 molar. Note that we are allowed to make this assumption of conservation of matter only because there is a 1:1:1 stoichiometric ratio between the reaction species, as confirmed by the question stating that each of the reactions are first order. But now we have an answer. Since we started with 0.01 molar of transcription factor beta, and now that we know that there's 10 molar at equilibrium, then based on the Intermediate Value Theorem, we know that yes, we will reach 8 molar beta at some point in time because 8 is in between 0.01 and 10. So now that we've determined that yes, there eventually will be 8 molar beta, our next step is to figure out how much time will it take for the cell to get to 8 molar. Well, how do we do that? Well, right now we have a set of differential equations that tells us the rate or how fast alpha, beta, and gamma are formed. But we don't have any function that tells us how much alpha, beta, and gamma we have at any given point in time. Essentially, we know how fast our transcription factor beta is being formed, but we never really know how much of it we have at any given time. For those of you familiar with differential equations, you know a secret. You know that the way we go from a rate to a value or function is by solving the differential equation. And if you think that's what we should do, you'd be 100% correct. By solving our differential equations, most simply done through integral calculus, we would be going from d beta dt to beta of t, a function that tells us the concentration of beta that we have at any given time. But if you even begin trying to solve our differential equations, you'll quickly start to realize that this is hard. Each of our equations has 4 different components that you have to deal with: alpha, beta, gamma, and time, as opposed to a simple x and y. And all these variables are dependent on one another. For instance, if we have more alpha, that increases the rate of formation of beta. So even though we really only care about transcription factor beta, we still need to keep in mind all 3 of these equations. This is what a lot of scientists and mathematicians call 3-dimensional dynamical systems, and they're incredibly difficult to solve. So what now? Well, some of you guys might have the idea to kind of brute force a solution computationally by using something like Euler's method, and by all means, go for it. But methods like that often aren't 100% accurate, and there actually is a clever way to solve this problem analytically, a way where we can get an exact answer. And this is where linear algebra comes in. Essentially, we will diagonalize a matrix by converting it into its eigenbasis so that we can reduce our dynamical system and be able to perform integration on our differential equations. So let's do that. We'll start by rewriting our system of differential equations into matrix form. We can simplify this further by abbreviating our vectors and matrices as dc dt, A, and C, respectively, with C meaning concentration. Note, this arrow is just a reminder that this is a vector. This notation is commonly used in linear algebra and physics. So now that we have our differential equations in matrix form, what next? Well, there isn't necessarily a simple and intuitive reason why you would know what our next step is, unless you're already familiar with linear algebra and differential equations. But basically what we want to do is convert our matrix A to its eigenbasis, because that will diagonalize our matrix and reduce our differential equations to just one variable each. So let's see this in action. We'll start by finding our eigenvalues of matrix A by using this formula. And by doing some determinant calculations and then some algebra, we find that the eigenvalues of this matrix are -5, -3, and 0. Then, using these eigenvalues and this formula, we can solve for our eigenvectors. For those of you who don't know, this formula is derived from the equality A times vector equals lambda times vector, which essentially states that a scalar eigenvalue can perform the same transformation on its respective eigenvector as an entire matrix, which is the essence of what an eigenvector is. But that's beside the point. After performing the calculations, you find that the respective eigenvectors are -1, 0, 1, 3, -2, -1, and 3, 10, 2. Now, we're able to start translating between our current standard basis and our eigenbasis. To do this, we will make a matrix S from our 3 eigenvectors. This is the matrix that will allow us to translate any vector from our eigenbasis to our original basis. To change basis in the other direction, from the original basis to the eigenbasis, we simply use the inverse matrix of S. We can solve for this inverse matrix through some long and complicated math that I won't get into this video, but just know that we're able to do it. Now that we have our matrices S and its inverse, we can translate our matrix A to the eigenbasis by performing matrix multiplication. Inverse of S times A times S. Matrix multiplication goes from right to left. So essentially what we're doing is transforming our change of basis matrix and then changing the basis of our transformation matrix from the original basis to the eigenbasis. So by performing this calculation, we now have our transformation matrix, but now in the eigenbasis. So we'll label this as A prime, with the prime symbol indicating that we're in the eigenbasis. Note, do not confuse this prime with meaning derivative, which is common notation used in calculus, but not really in linear algebra or this question. Notice how A prime is a diagonal matrix, meaning that all the values that are not on the diagonal are 0. This will be incredibly useful to us, as we'll soon see. As for the reason why converting to the eigenbasis diagonalizes our matrix in the first place, that's because our eigenvectors have now been set to be our basis vectors, which by definition move along only one dimension each. So now that we have A prime, let's rewrite our system of differential equations to the eigenbasis. So instead of this, we now have this. If we expand this out, we will have this set of differential equations. And if you're familiar with differential equations, then you'll know that these are now all solvable by simple integration. So through integration, specifically the method of separation of variables, we find that alpha prime of t is equal to some constant times e to the negative 5t. Through some additional substitution, we're actually able to figure out that our constant equals our concentration of alpha prime at time equals to 0. Thus, we can rewrite our solution to be alpha prime of t is equal to alpha prime of 0 times e to the negative 5t. Using the same mathematical procedures and logic, we can find the solutions of beta and gamma to be beta prime of t is equal to beta prime of 0 times e to the negative 3t, and gamma prime of t is equal to gamma prime of 0. So our next step is to plug in our initial values of alpha, beta, and gamma into our solutions. And you might be tempted to plug in 14.98, 0.01, and 0.01 because the question said that these are the concentrations that we started with at time equals to 0. But be careful. These values are in our original basis, while our differential equation solutions are in our eigenbasis. Thus, we need to convert our initial concentrations to the eigenbasis so that everything is in the same basis. Remember, to do this, we can simply use our matrix inverse of S, and by doing this, we get the following concentrations. So now our solutions to our differential equations read alpha prime of t equals 3.005 times e to the negative 5t, beta prime of t equals 4.995 times e to the negative 3t, and gamma prime of t is equal to 1. We're so close to being done. Remember, our goal was to find the amount of time it took for there to be 8 molar of transcription factor beta. Thus, what we need to do next is convert our solutions to our differential equations back to the original basis using our matrix S. And then we can solve for the value of t where beta equals 8. And if we do all that, we finally find that it would take 0.536 seconds for there to be 8 molar of transcription factor beta, and thus, that is how long it would take for gene X to be expressed. Now, I'll be honest, that might feel a bit underwhelming. We went through all that through biology, then chemistry, then differential equations, linear algebra, calculus, etc., all just to get one answer of 0.536 seconds. But the key takeaway that I want you to have from this video isn't to memorize the steps of solving this particular question. Truth be told, I'm fairly certain that over 90% of you watching this will never have to do this type of problem ever in your lifetime, even if you are pursuing a career in math or science. But what I do hope for you is to start internalizing the idea, the concept, that everything you learn, as random or useless as it may seem, all of it is connected in one way or another. Very little knowledge is ever useless. You'll find some way to utilize it one day. See, I find all of this to be absolutely beautiful because this single problem combines different minds, and it's highly representative of how science works in the real world. Biologists, physicists, and mathematicians aren't doing their own thing in their own rooms. Oftentimes, biologists need mathematicians to generate a computer program to solve biological problems. That's how NCBI BLAST works. And the problems that physicists solve might be utilized to better understand chemistry. That's most of quantum mechanics and atomic models. The important thing is to keep an open mind and to always be curious. Thanks for watching, and be sure to subscribe. This is my first video on this channel, and I hope to continue posting cool things in science and math. So subscribe for more. And as always, keep on appreciating.